My name is Scott Plafker, and my laboratory is at the Oklahoma Medical Research Foundation in Oklahoma City. The Oklahoma Medical Research Foundation, or OMRF, is a private nonprofit research foundation with about 50 independent laboratories. For the past several years, I've been interested in understanding how mitochondria are integrated with the cellular degradation machinery in the retinal pigment epithelium, or RPE, and how deterioration of these systems contributes to AMD onset. This tutorial will address the topic of mitochondrial dynamics. Specifically, I will review the major components and factors that regulate the capacity of mitochondria to undergo fusion and fission. In the second part of the tutorial, I will explore a hypothesis and present supporting data centered around the idea that disruptions in mitochondrial dynamics contribute to RPE loss and AMD onset. In an effort to keep this tutorial focused and brief, I will not cover in any detail the basic biology, architecture, and numerous functions of mitochondria, as these were the topics of comprehensive tutorials posted last year by Drs. Deb Farrington and Chris Kenny. A primary motivating factor for choosing the topic of mitochondrial fusion and fission for this tutorial is that the molecular details governing these processes have become apparent in recent years, and along with this information, it has also become apparent that deficits in mitochondrial dynamics are a common feature of numerous neurodegenerative disorders for which age is the leading risk factor. These diseases include Parkinson's disease, Alzheimer's disease, and Huntington's disease. Thus, a consideration of this topic in relation to AMD seems timely. It is, of course, well established that mitochondria serve critical cellular functions, including converting foodstuffs into energy in the form of ATP, storing and buffering intracellular calcium, sensing redox and proteotoxic stress, and under certain circumstances, initiating intrinsic programmed cell death, or apoptosis. But what does the term mitochondrial dynamics actually mean? Ironically, for much of the existence of modern cell biology, mitochondria have been depicted in textbooks as individual organelles with a kidney-like shape, as shown in the picture on the bottom left. This is despite the fact that the name mitochondrion, which was coined back in 1898, comes from the Greek words mitos, meaning thread, and chondrion, meaning granule. This name was chosen based on the various morphological configurations the organelle was observed to assume. These organelles, in fact, are constantly toggling between fusing with one another to form long networks and the process of fission, in which an individual mitochondrion is released by scission from the network. This equilibrium is illustrated in cartoon form at the top right of the screen. A depiction of these morphologies at steady state is shown in the photomicrographs on the bottom right, in which cultured human RPE cells, immortalized with telomerase, are labeled in red with the mitochondria-specific dye mitotracker. At homeostasis, the mitochondria are of intermediate length, as shown in the middle panel. Fused mitochondria have a more filamentous appearance, as shown in the right panel, whereas fizzed mitochondria appear vesicular or punctiform, as shown in the left panel. The dynamic nature of mitochondrial fusion and fission can be best appreciated by time-lapse video microscopy, as shown in the movie on this slide, taken from a review by Stephen Archer. In this video, cultured arterial endothelial cells containing a mitochondrially targeted photoactivatable GFP are excited with a laser, which generates GFP-labeled mitochondria. The GFP-labeled mitochondria first undergo fission and subsequently fuse with RFP-labeled mitochondria to create fused yellow mitochondria. Numerous other fusion and fission events are also visible in this 11-second movie. In my laboratory, we've been investigating these dynamics in mouse RPE flat mounts and have been able to capture short time-lapse movies from a transgenic mouse line that expresses a mitochondrially targeted GFP. For this movie, we took pictures every 15 seconds for 10 minutes. As with the cultured cells on the previous slide, we observed mitochondria toggling between states of fusion and fission. Interestingly, we observed most of the RPE mitochondria localized to the apical side of the RPE, unlike what is typically observed in fixed samples in which the mitochondria are predominantly distributed along the basal side of the RPE cell. Notably, this live cell imaging approach has the potential to determine how gene knockouts or transgenic expression of protein impacts mitochondrial dynamics. Let's now review the machinery that confer to mitochondria the capacity to fuse and divide. The enzymes that directly mediate both processes are GTPases, which catalyze the conversion of GTP to GDP. With respect to fusion, mitochondria have distinct GTPases to accommodate that they are double membrane bound, having both an inner and outer membrane. Fusion of the mitochondrial inner membrane is mediated by OPA1, whereas fusion of the mitochondrial outer membrane is mediated by mitofusins 1 and 2. As depicted in this cartoon, mitofusin dimers tether adjacent mitochondria and promote melding together of the outer membranes. Subsequently, through the action of the GTPase domain of OPA1 in the intermembrane space, the inner membranes fuse and the matrix materials of the two mitochondrion mix. Fission, on the other hand, is mediated by a single GTPase called DRIP1. DRIP1 is predominantly a cytosolic protein that is recruited to the mitochondrial outer membrane at sites of fission. This recruitment occurs at points of contact between the endoplasmic reticulum, or ER, and mitochondria, and involves the clustering of mitochondrial receptors for DRIP1. Two of the main DRIP1 receptors are FIS1 and MFF. Following constriction of mitochondria by the ER, an oligomerized ring of DRIP1 hydrolyzes GTP, resulting in scission of a mitochondrion from the network. The regulation of DRIP1 localization and activation is very complex, involving numerous kinases, phosphatases, and several ubiquitin and sumo enzymes, as depicted in the cartoon on the right. Further, DRIP-mediated fission is also modulated by the mitochondrial outer membrane proteins MID49 and MID51. The importance of mitochondrial dynamics to human health is perhaps best illustrated by the multiple diseases caused by genetic mutations in the fusion and fission machinery. As listed in this table, autosomal dominant missense mutations of mitofusin 2 cause Charcot-Marie-Tooth disease type 2A, a peripheral neuropathy disorder. Autosomal dominant mutations in OPA1 result in retinal ganglion cell death and blindness. In fact, mutations in OPA1 are the most commonly inherited cause of optic atrophy, and the protein derives its name from this phenotype. As DRIP1 is the primary mediator of both mitochondrial and peroxisomal fission, it is no surprise that DRIP1 null mice display embryonic lethality between days E9.5 and E11.5. Similarly, brain-specific knockout of DRIP1 is also embryonic lethal. A case report in the New England Journal of Medicine from Waterham and colleagues in 2007 reported on a newborn girl born at term with a point mutation in DRIP1 that abrogated the enzyme's capacity to properly oligomerize. She died at age 37 days. Her pathologies included microcephaly, abnormal brain development, persistent lactic acidemia, elevated plasma levels of very long-chain fatty acids, and optic atrophy and hypoplasia. She did not respond to light stimulation, had horizontal nystagmus with poor visual fixation, and her optic discs were pale and cupped. Such examples highlight the essential functions of maintaining the proper balance between mitochondrial fusion and fission. Also listed on this table are genetic disorders resulting from mutations in regulators of DRIP1, namely the kinase PINK1 and its substrate parkin. Mutations in the genes for both of these proteins are known to cause autosomal recessive juvenile Parkinsonism. These two proteins will be discussed in more detail later in the tutorial. Although to date specific genetic links between the mitochondrial fusion and fission machinery and AMD have not been identified, I think that investigating this aspect of mitochondrial biology could prove very fruitful for understanding the etiology of AMD. Several pieces of supporting evidence underlie this notion. First is the finding from Feher and colleagues that post-mortem histopathological analyses of AMD eyes showed dramatic disruption of the RPE mitochondrial network, including decreased numbers of mitochondria, decreased mitochondrial size, and a loss of cristae and matrix density, all of which are consistent with an imbalance in mitochondrial fusion and fission dynamics. These investigators also determined that peroxisome numbers in the RPE increase as a function of aging, and even more so in AMDIs. Thus, the decreased size of the mitochondria in AMDIs and increased numbers of peroxisomes are together consistent with an excess of DRIP1 fission activity. This interpretation begs the question of why then the number of mitochondria, like the number of peroxisomes, is not higher in AMDIs versus age-matched controls. One explanation is that many of the damaged mitochondria in the RPE cells from AMDIs have been triaged for degradation by mitophagy. More direct support for the imbalance in RPE mitochondrial dynamics in AMD comes from work done by Mike Bolton and Bernard Godley. Mike was kind enough to share this unpublished data with me for use in this tutorial. He and his team isolated and cultured primary RPE cells from AMD donor eyes and age-matched controls and discovered the following. First, there is a decrease in mitochondrial mass associated with aging and with progression of AMD as shown in the panel A graph. This confirms and extends the Feher findings from the previous slide. Secondly, RPE cells from AMD donor eyes show increased mitochondrial fragmentation, as illustrated in the panel B photomicrographs, again consistent with the hypothesis that fission activity is increased in the disease state. Panel C shows perhaps the most compelling evidence of increased fission activity in AMD donor RPE cells. This is a fluorescence recovery after photobleaching, or FRAP experiment, in which the fluorescent mitochondria are bleached with a laser, and the time it takes for the fluorescence in the bleached area to recover is measured. The longer the recovery time, the more fragmented the mitochondria, as the unbleached contents of the mitochondria adjacent to the bleached area are not efficiently mixing with the contents in the photobleached mitochondria. It is clear from the graph that the AMD-derived cells in yellow do not recover to the same extent as the age-matched, non-disease RPE in pink or RPE from young eyes in blue. When Bolton and colleagues used Western blotting to measure the levels of DRIP1, they found that the enzyme decreased with age, and further so with AMD. At first glance, this is perhaps the opposite that one might expect, since DRIP1 is the primary mediator of fission, and age in AMD increased the prevalence of Fis mitochondria in the RPE. However, an even more dramatic decrease was observed in the levels of the critical fusion factor, MFN2. Therefore, it appears that despite the decrease in total DRIP1 levels with age in AMD, the balance between mitochondrial fusion and fission is still tipped towards fission. A third rationale warranting the further investigation of mitochondrial dynamics in AMD is the large body of evidence compiled by many attendees of this conference that implicates chronic oxidative stress as a primary driver of age-dependent RPE atrophy. Sources of this stress and damage have been identified, such as cigarette smoke and photooxidized bisretinoids, but we still do not understand how these stressors damage RPE constituents, or equally importantly, why this happens as we age. This age-dependent accumulation of impaired mitochondria implies that in the disease state, RPE cells have largely lost their capacity to neutralize oxidative challenges and to effectively triage damaged mitochondria for elimination. A primary mechanism by which healthy RPE cells accomplish both of these tasks is through the mitochondrial fusion and fission machinery. In fact, mitochondrial fusion is a well-established mechanism for protecting cells against apoptosis. It confers such protection in at least two ways. First, it enables individually damaged mitochondria to meld with the mitochondrial network, mix and exchange contents, and thereby effectively neutralize insults that might otherwise trigger cytochrome C release and induce apoptosis. Secondly, the intermitochondrial membrane fusion mediator, OPA1, plays a critical role in stabilizing mitochondrial cristae, which not only improves respiratory efficiency, but also restricts cytochrome C release and ROS generation. In contrast to fusion, fission through the action of DRIP1 segregates damaged mitochondria from the network. These damaged mitochondria are either triaged for degradation by mitophagy, or under some circumstances can initiate apoptosis. Fitting with this second fate, DRIP1 has also been shown to play a coordinated role with the proapoptotic protein BAX to facilitate the initiation of the intrinsic apoptotic pathway. Consistent with the hypothesis that there is an imbalance of fusion-fission dynamics in favor of fission in the RPE during the later decades of life, Del Priore and colleagues demonstrated using flat mounts from human donor eyes that RPE cells undergo increasing amounts of apoptosis as a function of age, as shown in the panel A graph, and that this is especially true in the macula. Somewhat surprisingly, they also found that despite this enrichment of macular apoptosis, the density of RPE cells in this area remained unchanged with age, whereas it decreased in the peripheral retina. These data are shown in the panel B graph. The zones along the x-axis of this graph are depicted below the graph. These investigators interpreted this to indicate that peripheral RPE cells migrate to the macula as a compensatory mechanism to maintain macular RPE density. In keeping with the notion that increased fission in the RPE generates an abundance of damaged mitochondria, what is the fate of these organelles if they do not undergo the mitochondrial permeability transition and induce frank apoptosis? Healthy cells triage their damaged mitochondria for mitophagy, a specialized form of autophagic degradation. During mitophagy, fragmented mitochondria are engulfed by a de novo formed double membrane autophagosome that then fuses with a lysosome to become an autophagolysosome. Within the autophagolysosome, the lysosomal hydrolases degrade the damaged mitochondria and recycle the components for use by the cell. Recent studies have worked out some of the molecular details by which a damaged mitochondrion becomes a substrate for mitophagic degradation. This process is most often triggered by an irreparable loss of mitochondrial membrane potential and involves two proteins, PINK1 and parkin, mutations of which are known to cause familial Parkinson's disease. As an aside, mitophagy can also occur independently of parkin. The steps that are now well established for mitophagy involving both proteins, however, are as follows. The PINK1 kinase accumulates on damaged mitochondria and phosphorylates two substrates, one of which is the small protein ubiquitin that decorates the outer membrane of the organelle, and the second is the ubiquitin E3 ligase parkin. Phosphorylation of parkin activates it to extend the ubiquitin chains on the outer membrane. These phosphorylated chains of ubiquitin in turn become ligands for two different autophagosome receptors, optineurin and NDP52. There are actually 5 characterized receptors for autophagy cargoes, but Richard Yule's group published a manuscript in Nature this past August demonstrating that optineurin and NDP52 are specific redundant mitophagy receptors. Further, optineurin's role in mitophagy requires that it be phosphorylated by the tank binding kinase 1, TBK1. These two receptors, optineurin and NDP52, in turn recruit multiple autophagosome components including ULK1 and LC3. This chain of events thus leads to the engulfment of the damaged mitochondrion by the autophagosome. Interestingly, PINK1 and parkin are also known to control the stability of DRIP1, so future studies to determine the levels and activities of these proteins in aged human RPE should shed light on how both mitophagy and fission are regulated during aging in AMD. Notably, Esteve Rudd et al. Reported detection of parkin in the human retina by immunofluorescence analysis. So if the RPE cells in AMD have an excess of fission activity due to either an imbalance in the expression levels of the fusion and fission factors and/or because of chronic oxidative stress damaging mitochondria and sending them down the mitophagy pathway, one can envision that this would create an undue burden on the autophagy lysosomal system in these cells. It is well known through the work of many Beamer attendees, including Claire Mitchell, Sylvia Finneman, Mike Bolton, Jim Handa, David Zaks, and numerous others, that this major degradation system is severely compromised in diseased RPE cells. It is, of course, the lack of efficient lysosomal capacity that allows lipofuscin to build up in the RPE, due to the failure to effectively destroy the photoreceptor outer segments that are phagocytosed each day. Thus, it is my contention that an imbalance in mitochondrial dynamics, along with multiple other insults, exacerbates the compromised lysosomal system in RPE cells to promote AMD onset and progression. Excessive fission generates an abundance of damaged mitochondria that accumulate and further overburden the already deficient autophagy lysosomal machinery. This overburdening effect on the lysosomal system creates a vicious cycle in which the accumulation of lysosomal cargoes creates an ever-increasing cargo load that further taxes the system. For this reason, RPE cells from AMD donor eyes are loaded up with small broken mitochondria that are likely dumping reactive oxygen species into the cytosol and compromising overall RPE integrity and function. In this model, these accumulated compromised mitochondria lead to either frank apoptosis or severe dysfunction of the RPE cells, which in turn promotes AMD onset and progression. I would like to end this tutorial by mentioning that the cell biology of mitochondrial dynamics is a quickly expanding field of study as exemplified by the relatively recent discovery of mitochondrial-derived vesicles. These 70 to 150 nanometer vesicles pinch off from the mitochondrial network in a drip-independent fashion and deliver damaged mitochondrial proteins and lipids to other organelles, including peroxisomes and lysosomes. The PINK1 and Parkin proteins mentioned earlier in this presentation, as well as another Parkinson's disease-associated factor called VPS35, function in generating a population of these vesicles that provides a means of removing oxidized proteins and lipid from mitochondria for destruction by the lysosome. A cartoon of the defined steps in this process taken from a review by Heidi McBride's lab is shown on this slide. A combination of localized events, likely involving the accumulation of oxidized proteins within the mitochondria, a loss of mitochondrial membrane potential, mitochondrial chaperone saturation, and cardiolipin oxidation, cause an arrest in the mitochondrial import machinery. This arrest places the PINK1 kinase domain on the outer mitochondrial membrane, where it is situated to phosphorylate ubiquitin, as well as the ubiquitin-like domain of Parkin. Phosphorylated Parkin becomes activated as a ubiquitin ligase and is retained at the mitochondrial outer membrane. Activated Parkin, in turn, is required to generate the mitochondrial-derived vesicles, as are many other factors that remain to be determined. Whether and how these mitochondrial-derived vesicles contribute to mitochondrial integrity and function in aged RPE, and whether they contribute to overburdening of the RPE lysosomal system in AMD remain completely unexplored questions, with the potential to greatly advance our understanding of AMD etiology. So in closing, I'd like to offer a special thanks to the Beamer Executive Committee for giving me the opportunity to present a tutorial, and to my colleague Mike Bolton for kindly sharing unpublished data. I'd also like to emphasize my firm belief that the field of mitochondrial dynamics holds a treasure trove of information regarding the deleterious changes that occur inside RPE cells as a function of aging. I suspect fresh insights into AMD etiology will come from future investigations exploring this topic. And finally, for those individuals that would like to learn more about the mitochondrial topics that I've addressed in this webinar, I've included a few reviews to consult.